Hey everyone, and welcome back for another deep dive. Today we're getting microscopic and taking a look at enzymes.
Those tiny little workers that make life happen.
I like that. Tiny workers. Before we get too far, can you give us a quick refresher on exactly what enzymes are?
Well, think of your body like a really complex city, right? With tons of things happening all at once.
Okay, I can see that.
Enzymes are like the specialized workers that keep everything running smoothly and efficiently.
Uh-huh.
They're what we call biological catalysts,
Okay,
which means they speed up chemical reactions without being used up themselves.
gotcha. So they're like the ultimate multitaskers, making sure things get done quickly and efficiently.
Exactly. And they're not just limited to one location. They can work both inside and outside of our cells.
Wow. So they really are everywhere.
They are, and they're involved in pretty much every process you can think of, from digesting food to replicating DNA to fighting off infections.
Okay, so they're clearly super important, but how do they know what to do? Like, how do they choose which reactions to speed up?
Well, that all comes down to their structure. Enzymes are proteins, and each one has a unique 3D shape.
Okay.
This shape determines its function.
So it's like a key fitting into a lock. Only the right key will work.
That's the perfect analogy. Each enzyme has a special region called the active site,
Oh.
which is perfectly shaped to bind to its target molecule, which we call the substrate.
So the enzyme is like the lock and the substrate is like the key.
Exactly. And this is how enzymes are so specific.
So it's like a perfect match made in the molecular heaven. But how does this actually make the reaction go faster? I mean, it's all about energy,
Yeah,
right?
but you are. Every chemical reaction needs a certain amount of energy to get started, kind of like a kickstart. We call that the activation energy.
Uh-huh.
And enzymes work by lowering that activation energy barrier, making it easier for the reaction to happen.
So they're like giving the reaction a little boost.
Precisely. By lowering the activation energy, enzymes allow reactions to occur much faster and at lower temperatures than they would otherwise.
That's amazing. So they're like little energy savers.
You could say that. Now, when an enzyme and its substrate come together, it's not just a random collision.
Yeah.
It's more like a precise dance.
A dance.
Well, when the substrate enters the enzyme's active site, it might not fit perfectly right away.
I see.
So the enzyme's active site actually changes shape slightly to better hug the substrate.
Oh, so it's more dynamic than just a rigid lock and key.
Exactly. This is called the induced fit model.
Interesting. So they're not just workers, they're dancers too.
You got it.
Yeah.
But just like dancers, enzymes have their limits. They need the right conditions to perform their best.
Like what?
Well, temperature and pH are two big ones.
Temperature and pH. How do those affect enzymes?
Well, generally, increasing temperature speeds up reactions because the molecules are moving faster and bumping into each other more often.
Right.
But if it gets too hot, the enzyme structure can break down.
Oh, I see. Too much heat can be a bad thing.
Just like if you cook an egg for too long, it becomes hard and rubbery.
Yeah, that makes sense.
Yeah.
So what about pH?
Well, every enzyme has an optimal pH range where it works best.
Okay.
If the pH is too acidic or too basic, the enzyme can denature, which means it loses its shape and can't function properly.
So it's kind of like Goldilocks and the Three Bears. The conditions have to be just right.
Exactly. Temperature, pH, everything needs to be in the sweet spot for the enzyme to do its job.
Makes sense. And what about the amount of enzymes and substrates? Does that matter too?
Absolutely. The more enzymes you have, the more chances there are for substrates to find an active site and react.
So it's like having more workers on an assembly line. Things get done faster.
Precisely. But there is a limit. At some point, adding more enzymes won't speed things up any further.
Because all the substrates are already busy.
Exactly. We call this saturation. All the active sites are occupied and the extra enzymes are just sitting around doing nothing.
So it's all about finding that perfect balance.
You got it. And the same goes for substrate concentration.
Oh, so if you add more substrate, the reaction will speed up to a certain point.
Exactly. But eventually you'll hit saturation again when all the enzyme active sites are full.
So finding that sweet spot is crucial for both enzymes and substrates.
Absolutely. It's like a well-choreographed dance. If one partner is out of sync, the whole performance suffers.
I love that analogy. But sometimes, even the best dancers need a little help. What about those molecules that can help enzymes do their jobs better?
Ah, yes, those are called cofactors.
Cofactors, what are those?
They're non-protein molecules that bind to enzymes and help them function correctly.
So they're like the stagehands making sure the dancers have everything they need.
I like that. Some cofactors help bring the enzyme and substrate closer together, while others actually participate in the reaction itself.
Oh, so they're pretty versatile.
They are, and there are different types of cofactors. Some are inorganic, like metal ions,
Okay.
and others are organic, which we call coenzymes.
Coenzymes.
Yeah, they're often involved in transferring chemical groups between different enzymes.
So they're kind of like the choreographers, making sure all the dance moves flow smoothly.
Exactly. But of course, not every molecule wants to see the enzyme succeed.
You mean like those party crashers who try to ruin the fun?
You got it. We call those enzyme inhibitors.
Inhibitors, what do they do?
Well, they can either slow down or completely stop enzyme activity.
Oh, no. So they're like the villains in our enzyme story.
You could say that. There are different types of inhibitors. Some are competitive inhibitors.
Okay.
They actually mimic the substrate and compete for the active site on the enzyme.
So it's like an imposter key trying to fit into the lock.
Exactly. And by blocking the real substrate from binding the competitive inhibitor prevents the reaction from happening.
That's pretty sneaky.
It is. And then there are non-competitive inhibitors.
What do those do?
Well, instead of competing for the active site, they bind to a different part of the enzyme.
Oh, I see.
And this binding causes the enzyme's active site to change shape.
Hmm.
So even if the real substrate comes along, it can't fit anymore.
So it's like sabotaging the lock so that no key can open it.
Exactly. And these inhibitors have some really important applications in medicine.
Really? Tell me more.
Well, some medications work by acting as enzyme inhibitors. They can block the activity of harmful enzymes in the body.
Oh, wow. So we're using our knowledge of enzymes and inhibitors to design targeted therapies.
We are. It's pretty amazing what we can do with these tiny molecules.
This is all so fascinating. But before we go any further, I'm curious, how do scientists even study these processes? I mean, they're happening at a molecular level.
Right. Well, scientists have developed some very clever ways to measure enzyme activity.
Oh, really? Like what?
Well, one way is to track how quickly a reaction is happening, either by measuring the appearance of the product or the disappearance of the substrate over time.
So it's like timing how long it takes to bake a cake or how quickly the ingredients disappear from the bowl.
That's a great way to think about it. And to make sure their results are accurate, scientists always include control groups and repeat their experiments multiple times.
That makes sense. You want to be sure you're getting consistent results.
Absolutely. So that's a very basic overview of enzymes.
It is. And I'm already feeling like I've learned so much.
Well, me too. And we've only just scratched the surface. There's a whole universe of fascinating details and applications to explore.
Well, I can't wait to dive deeper. But maybe we should take a little break first.
Sounds good to me.
We'll be back in a flash to uncover more secrets of these incredible molecular machines.
Stay tuned. All right, so before the break, we were talking about how scientists actually measure enzyme activity.
Yeah, I'm really curious about that.
Yeah.
How do they track what's happening at such a tiny scale?
Well, imagine you're trying to figure out how fast a particular enzyme can break down a certain sugar.
Okay.
Scientists can set up an experiment where they mix the enzyme and the sugar together and then measure how quickly the sugar disappears over time.
So it's like watching how fast a cookie dissolves in milk.
That's a great analogy. And they can actually track that disappearance using different techniques.
Oh, really?
Sometimes they use a spectrophotometer.
A
A
what?
spectrophotometer. It's a device that can measure how much light is absorbed by a solution. And if the sugar absorbs light at a specific wavelength, then as the enzyme breaks it down, the amount of light absorbed will decrease.
So the solution will become less opaque.
Exactly. And the scientists can track that change over time to see how fast the enzyme is working.
That's pretty clever. But how do they make sure it's actually the enzyme doing the work and not something else in the solution?
That's where controls come in. They set up another experiment that's exactly the same, except it doesn't contain the enzyme.
Ah, so they have a baseline to compare to.
Right. That way they can isolate the effect of the enzyme and make sure any changes they see are really due to its activity.
Makes sense. So we've talked a lot about how enzymes speed things up, but what about those inhibitors? You mentioned they play a role in medicine, right?
Absolutely. In fact, some medications work by acting as competitive inhibitors.
Remind me what those are again?
Competitive inhibitors are those molecules that mimic the substrate and compete for the active site on the enzyme.
Oh, right. Like a decoy.
Exactly. They block the real substrate from binding, and that prevents the reaction from happening.
And how does that help in medicine?
Well, some medications used to treat high blood pressure, for example, work by inhibiting an enzyme that's involved in constricting blood vessels.
So by blocking that enzyme, the medication helps to relax the blood vessels and lower blood pressure.
That's exactly right. It's pretty amazing how we can use our knowledge of enzymes and inhibitors to design targeted therapies.
It is. What about those non-competitive inhibitors, the ones
Yeah.
that change the enzyme shape? Do they have any medical applications?
They do. Some antibiotics, for example, work by binding to bacterial enzymes and distorting their active sites.
So they prevent the bacteria from carrying out essential functions.
Precisely. And the great thing is that these antibiotics target specific bacterial enzymes that aren't found in human cells.
So they can kill the bacteria without harming our own cells.
Exactly. Pretty cool, huh?
It is. So these tiny molecules are playing a huge role in modern medicine. But I'm also curious about how enzymes are used outside of the medical field. You mentioned laundry detergent earlier.
Oh, yeah. Enzymes are used in a wide range of industries, from food production to textile manufacturing.
Really?
Yep. For example, in laundry detergents, enzymes like proteases and amylases help break down protein and starch stains.
So they're like tiny cleaning crews attacking dirt and grime at a molecular level.
Exactly. They make the stains easier to remove. And because enzymes are biodegradable and work at lower temperatures, they're also more environmentally friendly than traditional cleaning agents.
That's fantastic. So they're not only effective, but also eco-friendly.
Hmm.
What other industries rely on these amazing molecules?
Well, the food industry uses enzymes extensively.
Oh, really? Like how?
For example, the enzyme lactase is used to break down lactose in milk, making it digestible for people who are lactose intolerant.
Oh, that's right. I've seen lactose-free milk at the store. So those tiny enzymes are making a big difference for people with dietary restrictions.
They are. And in the brewing industry, enzymes are used to convert starches into sugars, which are then fermented by yeast to produce alcohol.
So those delicious craft beers I enjoy are all thanks to the hard work of enzymes.
It's amazing to think that these microscopic molecules are playing such a big role in our daily lives.
It is. And as scientists continue to discover and engineer new enzymes, the possibilities for their applications are pretty much endless.
This is all so fascinating. It's really eye-opening to see how these tiny molecules are working behind the scenes to keep us alive and functioning and to make our lives better in so many ways.
And we're not even done yet. There's still so much more to explore in the world of enzymes.
Okay, well, I'm ready for more. What other amazing feats can these enzymes accomplish?
Well, let's take a look at some of the cutting-edge applications of enzymes in research and technology. Things are about to get really exciting.
Okay, so we've talked about how enzymes are like little helpers in our bodies and how they're used in everyday products, but what about those cutting-edge applications you mentioned? What does the future of enzymes look like? Oh, the future is bright. Researchers are exploring some truly incredible things.
Oh, like what?
Like using enzymes to create biofuels, develop new diagnostic tools, even break down plastic waste.
Wow, that sounds amazing. Biofuels from enzymes. How does that even work?
Well, scientists are engineering enzymes that can break down plant material, like cellulose, into simple sugars.
Okay.
And then those sugars can be fermented to produce ethanol, which is a type of biofuel.
So we're basically using enzymes to turn plants into fuel. That's straight out of a sci-fi movie.
I know, right? And the best part is that biofuels are renewable, and they produce fewer greenhouse gases than traditional fossil fuels. So it's a win-win for the environment.
Okay, I'm definitely on board with biofuels.
Yeah.
What about those diagnostic tools you mentioned? How can enzymes help us detect diseases?
Well, enzymes can be incredibly sensitive detectors. They can be designed to react with specific molecules that are only found in certain cells, like cancer cells.
Oh, I see.
So imagine you have an enzyme that only binds to a cancer-specific molecule.
Okay.
Scientists can attach a fluorescent tag to that enzyme.
And when the enzyme finds its target, it lights up, signaling the presence of the disease.
That's incredible. It's like a tiny little beacon alerting doctors to the presence of cancer cells.
Exactly. And
Yeah.
we're really just scratching the surface of what enzymes can do in diagnostics.
This is all so mind-blowing. It seems like enzymes have the potential to solve so many problems.
They really do. And
Yeah.
here's another one. Plastic pollution is a huge issue, right?
Yeah, for sure.
Well, scientists are engineering enzymes that can actually break down plastics.
Wow, really?
Yeah. These enzymes could help us recycle plastics more efficiently or even break them down into harmless substances.
That would be a game changer.
So these tiny little molecules could hold the key to solving some of our biggest environmental challenges.
It's incredible, right? The future of enzymes is bright, and I think we can expect to see even more innovative applications in medicine industry and environmental science in the years to come.
This deep dive into the world of enzymes has been truly amazing. It's made me realize just how important these tiny molecules are. They're essential for life. They're used in countless everyday products, and they have the potential to change the world.
I'm so glad you enjoyed it. And remember, this is just the beginning. There's still so much more to learn and discover about enzymes.
Well, you've definitely inspired me to keep learning. Thanks for taking us on this incredible journey into the fascinating world of enzymes.
It was my pleasure. And to everyone listening out there, keep those brains buzzing and join us next time for another deep dive into the wonders of science.